The BMAC procedure pack consists of multiple single-use trays for the efficient and rapid preparation of bone marrow aspirate concentrate. First, let's identify the components in the BMAC 60 milliliter bottom tray. The BMAC bottom tray contains the following components: a bone marrow aspiration needle with a sharp and blunt stylet, a specially designed bone marrow aspirate filter bag for the pooling of the bone marrow as it's harvested, a blunt fill needle to cap the 60 milliliter bone marrow aspirate syringe, and a 30 milliliter ACDA vial. Next, let's identify the components in the BMAC sterile custom pack. The custom pack contains the following components: two 30 milliliter VAC-lock bone marrow aspiration syringes, a 60 milliliter syringe used to withdraw the pooled marrow from the filter bag, 3 disposable medicine cups for the heparin anticoagulant solution, ACDA, and local anesthetic used in the collection procedure. There's also betadine solution, a fenestrated drape, a skin marker, a scalpel, a spinal needle, and assorted supplies. Now let's take a closer look at the aspiration needle. Each needle assembly consists of an 11-gauge 10-centimeter long trocar needle and sharp stylet and accessory blunt stylet attached to a handle. A closer look at the needle tip shows the stylet extending beyond the distal hole of the needle trocar, and moving proximally 2 centimeters shows the 5 side holes of the needle trocar. Once into the marrow space, the sharp stylet is replaced by the blunt stylet in order to safely advance the trocar within the marrow space and reduce the risk of inadvertently penetrating the medial or lateral wall of the iliac wing. The 5 side holes allow more efficient aspiration of marrow while reducing the risk of damaging the aspirated stem cells. In the next few frames, we'll be looking closer at the kit components as we prepare these components for use in the bone marrow harvesting procedure. First, open the BMAC custom pack. Next, remove the components from the bottom tray and place them on the sterile field. These items include a bone marrow aspirate filter bag, an aspiration needle and blunt stylet, and an 18-gauge blunt fill needle for the 60 milliliter transfer syringe. Begin by closing the white slide clamp between the filter and the marrow bag. Using the 10 milliliter syringe, transfer the ACDA solution into a disposable sterile cup. Remove the red cap from the in port and place 4 milliliters of ACDA into the bone marrow aspirate filter bag for every 60 milliliters bone marrow to be collected. Ensure that ACDA coats the entire inside surface of the bone marrow aspirate filter bag. Now that all the sterile disposable components have been assembled, the next step is to rinse or coat all marrow contacting components and surfaces with heparinized saline. The recommended concentration of heparin used for harvesting bone marrow is 2,000 heparin units per milliliter of saline. 2,000 units heparin per milliliter is prepared by drawing up 8 milliliters of heparin solution, 5,000 units per milliliter in 1 milliliter vials, into a 30 milliliter syringe, followed by drawing up 12 milliliters of normal saline for a total volume of 20 milliliters of heparin solution at a concentration of 2,000 units per milliliter. This heparin concentration has been shown to be an effective anticoagulant for this procedure. Transfer the 20 milliliters of heparin solution into a disposable cup located in the sterile field. The first steps in the preparation for harvesting is to rinse all the aspiration syringes with the heparinized saline as shown. These steps will ensure that all marrow contacting surfaces of the syringes have been rinsed and coated with heparin. This is an important step that will reduce or prevent clotting of the harvested marrow. Following the heparin rinsing of the syringes, the aspiration needles need to be rinsed or coated with the heparin solution as shown. This is another very important preparation step which should inhibit or reduce the likelihood of clotting during the marrow aspiration. Finally, turn the plunger on the vac lock syringe counterclockwise until it is in line with the internal shelves. This will allow the physician to pull small volumes of bone marrow aspirate.